For hundreds of years, scientists and researchers have been on the hunt for a cancer cure, making incredible strides in treatment along the way. One of the main areas of focus has been on precision medicine, which has led to the development of targeted therapies that are more effective and have fewer side effects than traditional chemotherapy. Additionally, researchers have been working on developing immunotherapies that harness the power of the immune system to fight cancer cells like CAR-T cell therapy. These new treatments have shown promising results in clinical trials and continue to be explored. In fact, multiple measures show that CAR-T activity and its impact on patients have increased over the past 7 years. This transformative therapy has demonstrated the potential to disrupt cancer care, which has led to 10 CGT approvals for acute lymphoblastic leukemia, diffused large B-cell lymphoma, follicular lymphoma, mantle cell lymphoma, and multiple myeloma since 2017. Being known as the world's leader in frontier and emerging biology and first in market of the CN for cell and gene therapy industry, and now gaining the stringent ISO 9001:2015 certification. We will strive to consistently provide services that meet customer and applicable statutory and regulatory requirements for contract research and process development services for mammalian cells and their derivatives. Esco Aster will not sit idly. Learn how our groundbreaking manufacturing platforms for frontier and biology are involved in combating cancer in both cell and cell-free approaches. First on the list is using induced pluripotent stem cells for allogeneic CAR-NKT, CAR-T, and TIL cell manufacturing. These cells are then differentiated into the target cell types for CAR-NKT, CAR-T, or TIL cell therapy. As science continues to evolve, off-the-shelf natural killer cells can now be reproduced via iPSCs from healthy donors. At Esco Aster, we have a competitive manufacturing platform for cell reprogramming and banking derived from fibroblasts. These cells are then differentiated into the target cell types for CAR-NKT, CAR-T, or TIL cell therapy. Another bottleneck that Esco Aster will continue to address, and the most critical to a patient's outcome, is the vein-to-vein time. It is often referred to as the amount of time it takes for a patient to receive CAR-T therapy, from the time the T cells are collected through a vein to the time the modified T cells are infused back to the patient via another vein. For end-stage cancer patients, a 2 to 3 week timeline for this process can be problematic, as their cancer may progress during this time, making them ineligible for CAR-T therapy. Therefore, Esco Aster is making a significant step forward to reduce the vein-to-vein timetable. Using stable retrovirus-producing PG13 cell lines to transduce autologous T cells, which are licensed from Stanford and manufactured using our patented Tide Motion bioreactors, we eliminate supply chain bottlenecks. We are aiming for rapid cell processing using the fastest, automated, patented, linearly scalable microfluidics bioreactor with a 5-day turnaround time from receipt of leukopak to final frozen cryo bag. We also work on optimizing time-limiting quality control processes by offering 1-day rapid release testing, storing cells from early apheresis, and addressing an option for further in vivo cell expansion. We continuously develop novel approaches in protein or peptide transduced peripheral blood mononuclear cells and delivering cancer as a combination cell therapy or device for true point-of-care therapy. We are exploring direct-to-patient strategy that would allow drug therapies to be delivered and administered in the patient's home, as well as biological samples to be collected and transported from the patient's home akin to home dialysis. On cell-free approaches, Esco Aster is leaning towards the cell-to-cell transit system of exosomes from various potential payloads, from small molecules to small interfering RNAs. The combination of exosomes' inherent ability to carry various biomolecules and the effective delivery of these biomolecules into recipient cells makes them ideal nanoscale drug transport vectors, especially in the field of cancer treatment. In the field of cancer prevention, Esco Aster offers services with the use of lipid nanoparticles as an encapsulation tool in mRNA vaccine manufacturing, which has proven to be a useful platform to achieve a variety of clinical outcomes. In addition, personalized neoantigen cancer vaccine peptides are made possible using Esco Aster's synthesis and method development services for the purification of peptide components utilized in multi-epitope-based vaccine and neoantigen vaccine constructs. Esco Aster is driven to continuously provide innovative solutions to help beat cancer through the utilization of current technologies and building strategic collaborations with our services as the driving seat.